Okay, so this is where we left off last time. We're talking about, in addition to having to have transcription factors or proteins that bind to the promoter or switch regions of genes to turn transcription on and off, there's an awful lot of control that is at the level of chromatin. So whether a specific piece of DNA that has a gene is actually accessible or whether it's all messed up in what's called euchromatin, so it's like compacted. And we said that here you get a change in accessibility of the genes. So early on, no globin genes are expressed, and therefore these are, and it's because they are all in chromatin that is not accessible. The way we tell if a certain area of the chromosome is accessible to transcription factors is by using an enzyme called DNase that will break up DNA, and it is not sequence specific, so it would break anywhere in here, and you can get sensitivity there early on, and then you get a switch where later on, after 14 days of gestation, you get this gene is no longer accessible and these two others are, and that coincides with the time at which messenger RNA from this gene, the U gene, becomes less prevalent and goes away, and these two alpha globin genes, their messenger RNA becomes more prevalent. So when I was in graduate school way back when we were riding to work on dinosaurs and the earth was cooling and stuff, just volcanoes everywhere, there was a guy down the hall from us that was actually working on globin switching, but he was looking at transcription factors, and so there's multiple levels of control here. There's specific transcription factors that come on when these need to be activated as well, but then this stuff is later on, even after that. That was way back in the 1990s. And then in tissues that never express these, meaning something outside the red blood cell lineage, then you would have it compacted and not able to be transcribed, even accidentally, permanently. So now I have to, as a public service, we're this far in the course, I have to tell you that once again I lied to you, but it was, as usual, a lie of omission, where I have to tell the story that's easy to understand first and then the whole story later on. We talked about centromeres, and a centromere was a DNA sequence that binds to kinetochore proteins, and that's where the microtubules attach, right? So up here is what we're talking about. So you can see these are chromosomes, and the blue here is staining that will is a dye that stains DNA non-sequence specifically. So here, when the chromosomes are not compacted, you can see the blue is kind of diffuse, but there are certain spots where the centromeres are located, right? And so when you get into prophase, the chromosomes condense, getting close to metaphase, and then metaphase, the chromosomes all line up. Here are the spindles. So there's an antibody against tubulin in there that's green. There's a dye that just recognizes DNA that's blue. And then all of these rainbow colored guys here are different, are antibodies against different components of the kinetochore. So that shows where the kinetochores are attaching to the centromere sequence. If you look down here at the bottom, C. elegans and a number of other organisms have what's called holocentric kinetochores. So remember, like metacentric, submetacentric, acrocentric, telocentric is where the centromere sequence is located. Here you can see that the centromere-like sequence is located all along the chromosome. There's just little patches of it here and there, and so you can get attachment of the spindle microtubules anywhere along that. So just to let you know that that actually exists. What's going on? Oh. Well, yeah, if you're in line with that, move your stuff out of the way. So yeah, just pick it up and do the best we can. All right, so this allows interesting things to be done with C. elegans chromosomes that are primarily fun for geneticists to use as tools and not necessarily for happening in wild type organisms. So if you have remember when we talked about paracentric inversion heterozygotes and you ended up with like an acentric piece of DNA, that ended up being lost because if you don't have a centromere, you can't be pulled wherever you want to go. So with C. elegans, it's different. Now inversions still do the same thing, where if you get a recombination within an inversion, you get a gene imbalance, so everything that did recombination is dead. But if you were to get a piece that breaks off of a chromosome and doesn't include a translocation event onto another one, you can get a free floating duplication in C. elegans. So in other organisms, all the duplications that we talked about in chapter 8 have to go somewhere else in a chromosome because the chromosome is the carrier, and if a duplicated piece is just floating out in space in the nucleus, then it's going to be dead because there's no centromere and it can't get pulled where it needs to go. But with C. elegans, like if this piece were to break off right here, then it would be just fine. And so you can get what's called free duplications, which is a fun tool that we use sometimes. All right. So centromeres are at the at somewhere on the chromosome or all along the chromosome, depending if you have monocentric or holocentric kinetochores. Let's talk a little bit about the ends of the chromosomes. So telomeres are what protects the ends of the chromosomes, and you never know. I often learn things in class, and I was talking about those little plastic thingies on the end of shoelaces, and someone informed me that the correct name for those is aglets, and it is true. So now you've learned something truly useful in genetics. If only that today. You can be really smart when somebody talks about aglets now. So telomeres are exactly analogous to aglets, those little things on the end of shoelaces. What happens if you accidentally have your shoe untied and you step on one of those and it gets crunched? The end of it just unravels, right? These are protecting the ends from keeping everything from unraveling, and then it goes on down the shoelace and you're in trouble. Telomeres are on the ends of chromosomes, and so far, like early on, when I showed you the picture of telomeres, it was just like a little red spot on the end of the chromosomes, but now I'm showing you that telomeres are actually repetitive DNA. So usually they're a hexameric repeat, so you can see TTAGGG, right? It's just 6 nucleotides repeated over and over, and the opposite is on the opposite strand there. At each cell division, telomeres shrink, and they have to be maintained by an enzyme called telomerase that we will talk about in chapter 12. So coming right up. At each cell division, a little bit shorter, a little bit shorter, and a little bit shorter. So these are sort of like the time clock of the chromosome and the cell's life. There are companies now that are, well, they're not advertising so much anymore, but it was the hot thing maybe a year ago. Like, get your telomere length looked at, right? And you can see what's your age and what's your telomere age. Yes, more telomere length is good and less telomere length is bad, but as with most things, it's a lot more complicated than just looking at that. But once a telomere gets to a so short, the cell knows it and will stop dividing. Because if it continues to divide, it will continue shortening, then there won't be any protection and the chromosome can come unraveled. Yeah. Oh, hexameric, just Hexameric, just so six bases, yeah. A hexamer is a 6-base sequence, yeah. Yes. Yeah, you figured it out. All right. So, all right. What happens in cancer cells? So in cancer cells, did you guys talk about in cell biology, anybody's taken it the multi-hit hypothesis, or sometimes called the 2-hit hypothesis, but now the multi-hit hypothesis. So cancer is when cell division gets out of control and makes a big mass or a tumor. And then sometimes these cells can spread other places and seed other little tumors all over the place. So a bunch of things have to happen in order for that to occur. The first thing is that the control of cell cycle has to be off. Then also the mechanism, and we don't get into this in this class, but I assume you do in cell biology, the mechanism by which out of control dividing cells know that that's happening and enter into apoptosis or controlled cell death, that has to be lost too. So you have to lose control of your cell division, and your ability to kill yourself as a cell has to be gone as well. And a lot of ways, well, it goes both ways with this. So a lot of aneuploidy is involved in cancer cells, and that can start with losing telomeres, and the ends get ragged and you lose DNA. Another thing with telomeres that happens is that you will get overexpression of the telomerase enzyme that I promise I do show the mechanism of in the next chapter. So that normally when cells can only divide a finite number of times, then they have to stop. But if they have telomerase, that helps to do what's called immortalizing them, right? So it gives them the ability to keep on going and going and going because they can add to their telomeres. So there are a number of stem cells in your body, like the germline stem cells we've talked about. There's constant mitosis there. Because those cells are constantly dividing, you have to have telomerase present to maintain the telomeres. In your long bones, especially the femur, where the blood cells are, there's hematopoietic stem cells. They have to keep dividing and dividing and dividing. Telomerase has to be active there. But in most of your other cells, it's off. Now, if you get a wound or something and you stimulate cell division for repair, oftentimes telomerase will come on as well because it can sense that cells need to divide and divide and divide. In early tissue culture experiments, so taking cells out of a live eukaryotic organism and growing them on plates like you do in cell biology, and Dr. O'Neill does for her research, scientists found that there was a limitation in what's called primary cultures. So that's taking tissue directly from an organism, putting it on a plate. The cells can divide about 50 times, and that's it. They stop. This was referred to as the Hayflick limit because that was the scientist's name that did that. And so the ability to do tissue culture and use the cells that we're using in research now has depended on finding what's called immortalized cells. And these cells often have the properties of cancer cells where they've gotten the ability to divide over and over and over. Has anybody read The Immortal Life of Henrietta Lacks, that book? Yes? Everybody should read that book. Get it over Christmas. It's a long time been out, so it should be pretty cheap. This is about the first of these such lines called HeLa cells, which I've actually worked with in the past. They were recovered from a tumor from a woman who had cervical cancer. And so it's a long story, and I don't have time to go into it, but it's a really, really good book you should read. All right. So you do not have to memorize these. You do not have to memorize these. You do not have to memorize these. But you do need to know that each organism, although it has a different sequence, every chromosome in that organism will have the same sequence at the end of its telomeres. So all of your chromosomes have the same sequence at the end. The telomere sequence is not long enough, however, to allow non-homologous chromosomes to pair with each other. Remember, the X's and the Y's in males, for the ability to pair, they have to have that pseudoautosomal region, which is a much longer stretch of DNA at the end. All right, so this is how telomeres protect the ends of the chromosome. So the rest of the chromosomes going on down here, this is an electron micrograph. So you come up here, and then you get this T-loop. So it looks like sort of a lasso. This is what protects the end of the chromosome. Remember when we were talking about the structure of DNA, I said the bases are protected by being in the middle of the DNA double helix. A lot of nucleases, or enzymes that can destroy DNA, are exonucleases. These are ones that start from the end and move in. So we're trying to prevent the open end. So comes up around here. See that little thing right there, that little spot? Can everybody see that? So the whole thing here, this is a T-loop. This one is referred to as a D-loop, and that's actually where the end is going to tuck in there, as I'll show in the next slide. So this is the D-loop. Every telomere has a small single-stranded overhang of 3' end. And you can actually see, so this is the 5' end, and it goes around here to the 3' end that way. Because it's going the same way there, this sequence up here is exactly the same sequence as that one, and it can just tuck in there and base pair, and this sort of hangs out there on top. So you have the free 3' end tuck in there. In chapter 12, we see that even when telomerase acts to elongate the telomere, you always keep a free 3' end, and remember, it's to tuck in there, and that base pairing here locks it in place. The 6th edition purple book and the 5th edition, the sort of lavender light purple book, both have this figure correct. If you were looking at the older 4th edition, sort of the maroon color book, I don't know what the heck they did, but the sequence is messed up in there. So look at this figure, not the ones there. This is fluorescence microscopy of a metaphase chromosome spread. In this case, instead of the dye that glows blue that we saw earlier, there is a generic DNA dye called propydium iodide. I don't need to know that, but I think this one's called propydium iodide that glows red, that just stains the entire chromosomes. But then there is labeled DNA sequence probe that matches the telomere sequence, and it has fluorescence on it. So that's all these little dots here at the ends there. They appear yellow because the secondary antibody has a molecule on it that looks green, but when you have green and red overlapping, it makes a yellowy color. But you can see here then that the telomere sequence is the same at the end of every single one of these chromosomes, and that's the point of showing that. All right, I'm going to really quick before I go into chapter 12, we were talking about supercoiling, because I remembered the part for me today. So just like maybe I should make that. So just like DNA, this rubber vacuum tubing has a configuration that it wants to be in. It's springy and it has the lowest energy form. If I take it and twist it like this, I'm adding more turns, or you could say removing turns like DNA. I'm twisting it too tight. It wants to go back to the way it was. So if I'm holding it like this and it hangs loose, then I'm going to put the ends together here if I can. And you can't see this, but right here I have a line drawn with a Sharpie, so I can tell exactly, like if I'm going to turn it around once and twice and 3 times. So it usually takes 2 to get going, but I'm going to turn it this way once, and I'm holding it against it. If I turn it again, then it makes, see, it's got 2 supercoils, 1, 2, right? If I turn it around another turn, don't let go. Now it's got 3, 1, 2, 3. Every time I turn it around, it's going to do another one. So if I do 4, 1, 2, 3, 4, every single time. Now, what has happened to this? I'm not going to do it more times because it'll just fly loose if I won't be able to hold it tight enough. What's happened to this? Instead of being a circle that's just like flopping around and everything, it's gotten a lot more compact, and that's the point of supercoiling DNA. So DNA in eukaryotes wraps around histones, but even where it's not wrapped around histones, there's still supercoiling here, and in prokaryotes where there are no histones, supercoiling is very important. You will be able to tell later on in the lab when we run some DNA out on a gel about this. So if you take DNA and run it out on agarose gel, it will be pulled toward a positive electrode, and the longer the DNA is, the slower it goes. But the same piece of DNA, if it is cut so it's linear like this, it will run about the same spot. Let's say it's like 3,000 bases, it'll run about here. If it is supercoiled, because it's so tightened up, it'll run down here faster than that, than a linear piece. If it is not supercoiled and not linear, so a circle that's floppy like that, it runs way up here. So the same piece of DNA will run 3 different spots depending on the compaction, and supercoiling means more compaction there. So this is all well and good for prokaryotes, because whether you're talking about plasmid DNA, which is smaller pieces of DNA, or prokaryotic genomes, which are 5 to 10 million bases, it's covalently closed circular DNA. It's a circle where the ends meet. But our chromosomes are not like that, right? We just showed that even though they have that little loop at the end, our chromosomes are linear, right? So it's one long piece of DNA. So how does supercoiling work with linear chromosomes? In the nucleus, as in the cytoplasm, it's not just, as I've said, like a beach ball full of water. It's a very viscous environment. And so supercoiling works because there's not the ability of the DNA to unravel from one end, or uncoil from one end to the other, because it's so long. Use the garden hose example again. So if you take a garden hose and it's straightened out and you try to put it in a circle, remember I said it's like it's all kinked and twisted and you have to untwist it? Well, it's pretty hard to do that, isn't it? Because you're like trying to throw it like this, but because the garden hose is laying out on the ground, there's friction from the ground, so it's really hard for all those uncoilings to happen. So you have to kind of help it along with a lot of effort. And it's the same thing with the chromosomes here. The garden hose laying along the ground would be the linear chromosome, and the friction of the ground to keep it from uncoiling, and you have to use a lot of effort to throw the coils back out of it, is the same thing as supercoiling will stay in a local area, right? All right. So it's important, like these later chapters in the book, in addition to study questions from the book and the PowerPoint slides and the exam study guide, there's also videos in there, and they are fairly short videos. I'll try to remember to show them in here, but if I don't, and even if I do, they're good to go look. So the best way to approach this is to learn how everything works first, and then see the video, because it's somewhat hard to understand if you're just looking at the video first. So here we're going to actually find out what happens molecularly during S phase. And it's going to be exactly this. So this is a DNA polymerase enzyme. It is moving down a single strand of DNA, and it is using the bases sequence here to put new nucleotide triphosphates in there. So each of these here's a nucleotide with 3 phosphates on it, and each of these are getting added into there. And as they are, then the 2 phosphates are getting broken off and float away. All right, so where are we? We're right up here. So we've talked about the structure of DNA, chapter 10. Chapter 11, we talked about how DNA gets packaged. If you are not eventually going on to take molecular genetics, and most of you aren't because it's not going to be offered until the spring of 2021, there is a really good treatment at the end of chapter 11 that we do not have time to get to in here that talks about the molecular mechanism of homologous recombination. That would be really good after the semester is over for you guys to look through and read to get a good understanding, but we don't have time to do that in class. So now we're going to do DNA replication. So you have to be able to take one double-stranded DNA molecule and make 2 double-stranded DNA molecules out of that. So you're copying from 1 strand to 2. This has to be faithfully executed because, as I said before, if you make too many mistakes, then you're going to have a lot of those mutations are going to be bad for the organism, and therefore the population suffers. But it can't be too perfect, because if it is perfect, then you never get any mistakes or base changes, which are the allele changes that are required for evolution to occur. All right, so how does DNA replication work? At the beginning of chapter 10, I showed a quote from Watson and Crick's famous Nature paper where they said, now that we know the structure of DNA, it's obvious how that DNA can be copied. Because if you know the sequence on one strand, you can figure out the sequence on the other strand of DNA. Because across from every A is going to be a T, across from every G is going to be a C, etc. But exactly how does that work? 3 models were proposed. The first is conservative replication. Why is this conservative? It's got nothing to do with your political bent. It's that the original DNA after replication is conserved. The 2 strands here would come apart, be copied, and then you'd get a new copy here. So in this case, blue is the original, and this orangey red color is the copied DNA. So at each replication, at the first replication and at the second replication, the original DNA would be conserved in its entirety. The whole molecule would be together. The second is dispersive replication, where throughout the molecule you would have random old and new parts of DNA. And the third model, which actually turns out, spoiler alert, it turns out to be correct. The correct one is semi-conservative. So in this model, the two DNA strands separate, and then each blue strand that's old original DNA gets a second red strand put on it so that you have this. It's different from dispersive because some areas you have two new strands, or two old strands, somewhere you have two new ones, somewhere you can have half old and half new, etc. But here, the entire length of the DNA molecule, one strand is all old DNA, and one strand is all new DNA. All right, so how does this work? Another fun experiment. This one conducted by Messelson and Stahl. And so we can see, like 1958. So we're a few years after 1953 when the structure of DNA was first reported. This is another really cool experiment using a tracer similar to the Hershey and Chase experiment, where they used radioactive isotopes of phosphorus and sulfur. Only in this case, we're going to use a heavier isotope that is actually stable. So normally, nitrogen is number 14 if we had a periodic table. There's 14, there's 7 protons, 7, well, I'm not a chemist. Sorry. So you 7 protons and 7 neutrons for 14 total, and then you would have the cloud of electrons after that. Sorry. I need to get a periodic table that I can just bring in so I can look at it. Anyway, this 15N or heavy isotope has a molecular weight of 15 because there's still 7 protons, but there's an extra neutron. So there's 7 protons and 8 neutrons. Now you get an atomic number of 15. But unlike the 32P and 35S, this is stable. It's not going to disintegrate. Instead, the way we're going to figure out what goes on with this is actually using the fact that this 15N nucleus has more mass than a 14N nucleus. It's pretty amazing, like the difference of 1 proton in the nucleus is, and we're going to be able to tell, it is going to make a difference. We're going to do a labeling experiment just like before. So E. coli are grown for many generations in the presence of 15N, so that their DNA bases, which have those nitrogens in, as you've seen, all the nitrogens are the heavy kind, the 15N heavy isotope. Then they spin down the cells, wash them, and move them to a new medium that has 14N, or the light isotope of nitrogen. And they can calculate one generation, a second generation, and a third generation, etc. Because just like they knew the T2 bacteriophage life cycle in the Hershey and Chase Waring blender experiment, here they know exactly how long each generation takes, and they can take samples at each of those. All right. So at each generation, you're going to have the new DNA is going to be made with the 14N light nitrogen, and the old DNA is still going to have the 15N heavy nitrogen. So how do we tell the difference? We use a technique called density gradient centrifugation. So originally up here, all of your DNA molecules are mixed in a tube with a solution that is capable of forming a density gradient. What does that mean? If you had no DNA in these tubes and you spun the tubes for a very long time at a very high RPM, and we're talking about like a continuous spin for like 2 or 3 days, then you can see here that, don't pay attention to this, but look at the light coloring up there and the dark coloring there. The solution would naturally make a gradient where toward the top of the tube the solution would be less dense, and at the bottom it would be more dense. And once that gradient sets up, the gradient stops. And you're spinning your solution for so long that any other molecules, like the DNA that's in there, after a while will stop at the spot where it matches the density of the solution. This is different than all the centrifugation you guys have probably ever done, in which you're just spinning something in a centrifuge and what you want is where? It's on the bottom, right? Because you're usually spinning in water, and whatever you're spinning down, like cells, usually are heavier than the water and they sink to the bottom. Here, the lighter DNA would stop at one spot and the heavier DNA would stop at another spot. Because the gradient solution below this 15N spot is more dense, no matter how long you spin it, once you get to this point, you could spin it for a year and it would never change. The items stop at each density. And you could take 14N, you would know exactly where it is. You would take 15N, you would know exactly where that is too. So you could do an experiment with only 14N and another experiment with only 15N. All right, let's look here. At the beginning of the experiment, when you've just been growing it in 15N forever, if you would spin that down in a density gradient, all the DNA would be down here at the bottom because it's all heavy. Transfer the cells, wash away the 15N media, make them up to in 14N media, allow the cells to go one generation. So half the DNA would be 14N and half the DNA would be 15N. And what happens is they found the band exactly halfway between there, because it's no longer as heavy as this, because half of the one whole strand of the DNA is now made with the light 14N. It's not up here either, because it's one strand is with the 15N, so it's right in the middle. And this is the cool part. Then when you go from generation 1 to generation 2, spin that down, you see for the first time a band up here at the top. Because if you look here, after one generation, the original DNA, these two strands come completely apart, and each one then gets a new strand on. So each piece of DNA has one strand heavy and one strand light. It's exactly in the middle. Now these separate. The blue strand out of here gets another new strand, but it's still heavy. So this DNA here is going to still be about the middle of the weight. But the new DNA, the red strand here, it's already 14N. And so the new strand that goes with that is 14N too. So now you get a strand that's entirely 14N, and boom, it pops up here. See how that works? And then each generation, these two original guys, the original guys here that still have the heavy strand, it'll always be around. But half of these are going to become light, and all of these are going to stay light because they don't have any heavy in at all. So what happens is every generation after the second generation, the band at the light part will have more DNA there, and the band right in the middle will be less and less and less and less. Yes, even after 1,000,000 generations, you'll still have the original DNA there somewhere, but it'll be so diluted out that it'll be gone. And at some point, those original strands aren't going to be, are going to be not even a part of it anymore, because those cells will have died off and will be gone. So you would get more and more and more here at the light area, and that will slowly disappear until it's all gone. Make sense? Really makes sense? Well, let's see if it does. So that showed that semi-conservative replication is what actually happens. So down here, this is just so you can see the light area and the heavy area and think about what happens. But let's say, so what would happen if replication was conservative like this? At the beginning, everybody's still going to be all 15N heavy nitrogen because that's all it was. But after one generation, you have this heavy nitrogen replicated into these two. And remember, the new DNA, the red DNA or the orange DNA, whichever you want to say, it's halfway in between, is all light nitrogen. So what would happen at this spot? What would you expect to see there if conservative replication had actually happened? Right, both right. It was the top, you would see one band at the bottom and one band at the top. Why would there be no band in the middle? Because it's either all heavy or all light, because the heavy DNA was conserved after the replication was over. And then, so what would happen when you go, so in this one, after one generation, half would be here, the light, half would be there, heavy, because that's what this is. So in subsequent generations, what would happen? It would eventually all be in the light DNA, because the heavy DNA after a while would be again diluted out to the point where there wouldn't be any there for you to see. But would you ever see a band in the middle? No. So what would happen would be that every generation the light band would get darker and the heavy band would get lighter until it's all light band and no heavy band. Question? All right. So what would happen instead if you were having dispersive replication like this? Right. It would all be in the middle. It would all be in the middle, same as this, right? After the first generation. So what would happen to the second generation? That's the same. How do we know it's not, how did they know it wasn't dispersive then? What would happen, so at because it's semi-conservative, at the second generation, you get the first appearance of the 100% light DNA. If it's dispersive like this, what would happen at the second generation? It would all still be as one band in the middle, but where would that band be? It would be a little bit moving up toward the top because there'd be more light. And so each generation then for dispersive, that band would still be in the middle, but it would move more and more and more and more closer to the top until eventually it would be right at the top band because, again, all the heavy DNA would dilute out. I'm always just amazed when I talk about this experiment that you can centrifuge through a density gradient and tell whether this DNA has one extra proton or not. It's or one extra neutron or not. They're not a positively charged molecule. All right, so let's talk now about the mechanisms of replication. So we're going to talk first about circular DNA. This is the DNA that's in the nucleus of prokaryotic cells as chromosomal DNA, and also DNA that is plasmid DNA, so smaller circles of DNA that can be exchanged between bacteria. So these all use the theta replication mechanism. There's another replication mechanism discussed in your book that's called rolling circle, but we don't have time to get to that, and so we leave that. You'll talk about that in microbiology class when you take it. The reason why this is called theta replication is because after it's about half over, if you would take this DNA molecule and turn it so that this part here is up at 12 o'clock, it would look like the capital Greek letter theta with the little curly Q through the middle. So here's how it works. There's an origin of replication here. That's where it's all going to start. There's sequences, DNA sequences at the origin of replication that are going to attract the proteins that are required to carry out this process. And so once this gets started, the DNA separates, and you can see that each of the gray strands here comes apart and is single-stranded DNA, and then new DNA is added to each of those gray single strands. This is called a replication bubble because it opens up and the bubble gets bigger and bigger. Each replication bubble contains two of what we call replication forks. So that's here and here. They're called a replication fork because see, it's one here and then the DNA forks out as it separates into single strands. And there is a replication fork here as well. The two replication forks, as indicated by these arrows, are moving away from each other. So more and more of the double-stranded parental DNA, the gray strand there, is separated into single strand, and you get new DNA, sort of the whatever that maroon color strand is right there, until you get down to the very end here, and you'll end up with two circles. These circles will be catenated or joined like this. The reason for that is because the DNA is covalently closed, and so when it all unravels, you'll get two circles that are connected like the Olympic rings. And so the action of a topoisomerase 2 is needed. So a topoisomerase 1, remember, just breaks one strand and rotates it around the other one. Topoisomerase 2 can break both strands and pass a double-strand DNA molecule through it. And so it can break like here and then separate them and they come back together. So now you go from one DNA strand to a second DNA, one molecule to two here, and then cut them apart. You can actually see this in electron micrographs. So this is plasmid DNA because the millions of bases in the chromosome would be just way too much to see. The origin of replication had to be right here in the middle because now you can see one fork moving down that way, another fork moving up here. Here's why it's called theta replication. You're about halfway done. Here's even more, and then here are the two circles. There's one, there's the other one, and they are joined together and have not been separated by the topoisomerase 2 yet. In prokaryotes that use this method, there's only one origin of replication. The DNA separates at one spot, and then you get two replication forks, one moving away from each other in opposite directions until this fork meets this fork here, and that's when you get this and you're done. So prokaryotes have a single origin of replication. Eukaryotes have multiple origins of replication, and also they are linear. The reason you need multiple origins of replication is because eukaryotic chromosomes are much longer than prokaryotic chromosomes. And also, eukaryotes don't have a trick that prokaryotes use. So we'll see this in a second, but prokaryotes actually start replicating their genome again before the first replication is finished. Eukaryotes can't do that. Right? So you have multiple origins of replication all along the chromosome. All of these fire or start replication, if they're going to, during S phase. Here you have these two starting and they move together. Just like in prokaryotic, you have two replication forks, one here and one here, for each replication bubble. That's the whole thing. Each replication bubble contains two forks, and they move apart, getting closer and closer. Now this one's got a late start, but there it goes. And so each of these replication forks are moving toward each other until the replication bubble eventually pops. Like when these two come together, now it's all open. These two come together, now it's all open, right? And you get two new DNA molecules there. It is very important for replication to take place only during S phase, because if you're undergoing cell division, after replication you go into G2, then you go into M phase for either mitosis or meiosis. If replication is still going on at that point, you're going to have multiple DNA molecules and they're going to break apart, and it's going to be bad news. So to prevent replication from happening at any time other than during S phase, there is a licensing mechanism that occurs. So there is something called replication licensing factor. It's actually a complex of multiple proteins that we're just calling licensing factor now, so you don't have to remember all the different names of them. This will bind to origins of replication during G1 phase. Licensing complex can only bind new during G1 phase, not during S, G2, or M phase. Then when you move to S phase, only origins of replication that possess the protein licensing factor are able to fire or start replication. So if you did not, I mean this one is starting here, but let's pretend that this one did not get licensing factor in G1, it would just still be here and this would never happen. Uh-oh, the replication would never happen there. If it didn't get the licensing factor during G1, during S phase, you could never have replication starting. But it's not a big deal. You're like, oh my goodness. In prokaryotes, if that were to happen, then there's only one origin of replication. Replication just wouldn't happen. But here, there are so many origins of replication all along the chromosomes that it's not a big deal. And so if this origin of replication never fired, then this one here going that way would replicate the sequence, or another one over here to the right going that way would do it. But this mechanism ensures that replication only can happen during S phase and can only happen once. So once these are copied, like there's going to be an origin of replication here and one here on the two new strands. But because the licensing factor protein complex can only bind during G1 and now we're in S phase, can't bind and we're done. Good, good? All right. So I'm going to start talking, we have a few minutes left, I'm going to start talking about the mechanism of DNA replication so that on Tuesday I can come back and we're going to go over this again so that so you get it twice. See how that works. All right, so here is our deoxyribonucleotide triphosphate. So here's the deoxyribose. It's deoxy because the 2 position does not have a hydroxyl group. It's got a base here. This is not to scale, obviously. And then it's got a triphosphate here. It takes lots of ATP energy to build these. And these 2 bonds have a lot of energy in them, especially this one. What's going to happen is that as you add one of these nucleotides to the 3' end of a chain, the 3' hydroxyl that's here at the end of the chain is going to attack this bond, leading to a joining right here. They will have the phosphodiester, so this oxygen is going to be connected to the 5'. This oxygen, as we'll see, will be connected up to the 3', and this pyrophosphate will fall off. That releases some energy, and then it will spontaneously decay into 2 inorganic phosphates when this bond breaks. That releases an absolute ton of energy. That's the energy that drives DNA synthesis. It's built into the deoxynucleotide triphosphates during their synthesis by burning ATP, and then this energy that was added by having 3 phosphates on there is released in a big way during DNA synthesis, and that's what drives the reaction to completion. All right, so this DNTP, or deoxynucleotide triphosphate, is this guy down here. So here's the base, 1, 2, 3, 4, 5 prime carbon. These 3 things here are phosphates. When it comes in, the 3 prime hydroxyl on the chain is going to attack right here and lead to the joining of the new nucleotide to the old one through the phosphodiester bond. This is the pyrophosphate that comes off, and it decays to make 2 inorganic phosphates, driving the reaction to completion, and that's what you get here. As I'll say again next time, which nucleotide is chosen is not determined by the DNA polymerase. The DNA polymerase cannot read the base sequence. Instead, it's the pairing of the bases that does that. When this C comes in and base pairs with the G like that, only then does this 5 prime phosphate get close enough to the 3 prime hydroxyl for the reaction to occur. If it was a different base, in almost every case, with some exceptions where there's accidents, it never gets close enough for this reaction to occur. So it is, and I'll show it again next. It's the base pairing that determines what happens, not the polymerase. So cute. So it's I mean, this is going to be for RNA because it's got uracil, but you know, I want to be adenine, so I can be paired with T. Doesn't work so well. All right. So I'll see you guys at the end. Somebody's coming in at 10:50 today, but so anytime after, like, you know, 11:10 or so, I'm able to meet with anybody who wants to come in and see me. Other than that, I'll see all of you guys at the lab time that are the Thursday lab people.